This is the third set of notes on Chapter 9, Genetics. This time we're going to be talking about gene expression and pedigrees. So gene expression is the process that that talks about how the gene codes for whatever the trait happens to be. Okay, the book definition is the process whereby genetic information flows from genes to proteins. Okay, we're looking at the genotype and how it produces the phenotype. The gene expression is regulated by turning genes on and off as needed, and let's think about why that would be necessary. Okay, every cell in your body has the same chromosomes, the same DNA as every other cell, but every cell doesn't have to do the same jobs. You have cells that are differentiated or that are specialized to do different kinds of jobs. You have nerve cells that transmit impulses. You have muscle cells that contract. You have gland cells that produce hormones. The muscle cells don't have to produce the hormones that are produced by the gland cells, and the gland cells don't have to contract like the muscle cells do, or they don't have to transmit impulses like the nerve cells do. And so you need to be able to turn on the things, the genes that are needed by that particular kind of cell or that tissue, and turn off the genes that are not needed by that. Okay, and so that's what we're talking about gene expression. In fact, that gene expression is regulated by the organism or regulated by the cells. Now there's a difference in gene regulation between prokaryotes and eukaryotes, and we're going to talk about prokaryotes first. So we're going to look at prokaryotes first, and then we'll talk about eukaryotes. Prokaryotes, like E. coli, have been studied extensively, okay, and that's where this process was first determined or first discovered, first identified in E. coli and organisms like that. They have these clusters of genes in their chromosome that have the clusters of genes that have related functions, and these clusters are called operons, okay. They have a control sequence that includes a promoter and an operator, and then they also have repressors. Some operons are ones that are repressible. That means you can turn it off until you need it. Others are inducible, meaning that they can be turned on when needed rather than turned off when they're not needed. And so it just depends on what the operand does and what the needs are of that particular cell. Now here are some vocabulary terms about what I just mentioned. Okay, a control sequence is the section of DNA that helps control the operon. Okay, it's going to be where the instructions for taking care of those particular set of genes is located. The promoter is where the RNA polymerase is going to attach. That's part of the control sequence. The RNA polymerase, remember, attaches to the DNA, and then it produces that messenger RNA that is needed to make the protein that is needed for the trait that is going to be expressed here. The operator, also part of the control sequence, is the on-off switch. It's what says, okay, it's time to make this protein or it's time to not make this protein. And then a repressor, if there is a repressor, binds to the operator, it attaches to the operator, and physically blocks the RNA polymerase. So basically it stops the RNA polymerase from attaching or from transcribing the DNA, and so it stops things from happening. We're going to see how that works. So here's a commonly studied operon called the lac operon. The lac operon is responsible for making genes that can utilize lactose. Lactose, remember, is the sugar that is found in milk. Okay, and let's say you're an E. coli residing in your stomach, and if you're not drinking milk or eating yogurt or something like that, you don't really need to be able to break down lactose. It's not really important, and so that would be turned off because lactose is absent. You don't need to be making lactase or the lactose utilization genes. You don't need to be making that at all. It's why waste the energy to make something that you don't need. Okay, and so in the case of the lac operon, it is what's called a repressible operon. It is turned off. It has a repressor there. It's attached to the operator sequence that prevents the RNA polymerase from attaching and transcribing those lactose utilization genes. Okay, it is repressed. It's turned off when lactose is absent. But let's say you just drank a glass of milk, and so now your E. coli has lactose available, and it's going to need to break it down because lactose is a disaccharide. It's going to need to break it down to get those monosaccharides to do whatever job the cell needs to do with those, whether it's producing energy or whatever. And so now you need to turn that off. So what happens in the case of the lac operon is when lactose is present, it binds to the repressor, and that changes the shape of the repressor and makes it turn loose from the operator. And then the RNA polymerase can attach to the promoter and can be transcribed into the messenger RNA, which can then be translated into the enzymes that are needed to utilize lactose. Simple process. Some operons are repressible, like this one. Others are inducible, and we're going to look at some of those in class. But some of them are inducible, and that means you can turn them on when you need them, rather than turning them off rather than leaving them off and turning them on when you need them. They can leave them on and turn them off when you don't need them. Now in eukaryotes, it's different, okay? Remember, multicellular eukaryotes have differentiated cells to produce a lot of different cell types, okay? Each different kind of cell has a different kind of program of gene expression depending on the developmental signals that stimulated the production of that kind of cell during the embryonic development, okay? And the differences between the cell types are because of this selective gene expression. And there are a lot of different things involved in this. It's a much more complex process in eukaryotes than it is in prokaryotes like E. coli. There are a number of factors that can affect this selective gene expression. One factor is called DNA packing. If the DNA is tightly wound up, it's going to block gene expression because the RNA polymerase is not going to be able to attach to the DNA because there's not going to be an open space for it to be attached there. Another factor that can affect this expression is called chemical modifications, okay? And this is when you have additional chemical groups, functional groups can be attached to some of the amino acids or nitrogen bases that can also prevent gene expression, or in some cases enhance gene expression. And so depending on what kind of cell it is and what needs to be done, those modifications can either allow that to occur or prevent it from occurring when it's not needed. One thing that's been learned in recent years is something called epigenetic inheritance. And this is inheriting traits that are transmitted by mechanisms that aren't involving the DNA sequence, and some of those are related to chemical modifications in the chromatin, because those additional functional groups that can change or influence the expression of genes. These can be inherited from parent to child. So it can affect gene expression. It can help explain differences between identical twins. There are a lot of different things that can be involved there. It's a pretty complex process, but very, very interesting, and one of the most recently widely studied fields in genetics. There's a video that I'm going to have you watch about epigenetics that's really interesting, and it'll be an assignment that you'll need to watch. It's from the SciShow series on YouTube, and I'll need you to do that, and we're going to have a quiz over it in class. So I'll tell you about that in class. Another thing that you need to know that about gene expression is something called X-chromosome inactivation. In female mammals, since you've got 2 X chromosomes, and remember you have to have at least one X chromosome to survive. Males get along just fine with their 1 X chromosome. Females have 2, but you only need 1, and so most in the body cells, one of those X chromosomes is chemically modified and becomes inactive and produces something called a bar body. Which one is turned off depends on lots of different things. It's just in the embryo, it's just a matter of chance which ones get turned off. But from those stem cells in the embryo, where the one gets turned off and the other one does not, all of the daughter cells from that embryonic cell will have the same copy turned off. And so sometimes you end up with patches of cells that have 1 X chromosome turned off, and another patch nearby will have the different X chromosome turned off. Here is an example of X chromosome inactivation in tortoiseshell cats. When we talked about cats in the previous notes, I told you that the inheritance of cat color was different than what it showed there in the examples that we were giving. And cats' fur color is on the X chromosome. Okay, and so here we have an allele for orange fur and an allele for black fur. Okay, and then you have those are in the somatic cell. And so in the early embryo, you'll end up with a cross between those two things. And then as the embryo is developing, randomly groups of cells will turn off 1 X chromosome or the other X chromosome. And so if you have the a cell population that has the orange X active and the black X inactive, then you'll have a patch of orange fur like you have here in this tortoiseshell. Or if you have a patch that has an inactive X with the inactive orange allele and the active black allele, you'll end up with patches of black fur, and this produces something called a tortoiseshell cat. Another thing that you might know about cats is cat color is calico cats. Calico cats have 3 fur colors, and the only way they can have that is if they have 3 X chromosomes. And so you'll have, if you have 3 X chromosomes in a female, then that's okay, because again, you only need 1. You've got 1, the others will get turned off most of the time, but you have patches that different ones are turned off. And so most of the time when you see a calico cat, you'll know that it would be female, because that's the most common way for it to happen. But you'll also know that that female has 3 X chromosomes, so it's the result of a type of non-disjunction, like we talked about with meiosis. If you ever see a calico cat that is male, you'll know that that male is also infertile or sterile, because he's got 3 X chromosomes as well, and that's going to be kind of like Klinefelter syndrome in humans. It's another non-disjunction disorder where they're going to have two more than they need X chromosomes with that Y chromosome, and they're therefore going to be infertile. Now, when you're looking at organisms with longer lifespans and you're trying to figure out the occurrence of inheritable traits over generations, it's kind of difficult to do in humans and some larger animals. It's kind of difficult to do those test crosses. And so oftentimes what we do is make a diagram called a pedigree. Okay, this shows the occurrence of those traits over a number of generations. And so there's some things you need to know about pedigrees. We'll do an activity in class about pedigrees. They're not really difficult to understand, but you need to have a little bit of background about them to begin with. In the pedigree diagram, the circles represent females and the squares represent males. The colored in shapes have the condition that you're looking at. It doesn't say whether it's dominant or recessive, it's just that it's present there. Half-colored shapes when they're present. This particular pedigree doesn't have those, but some of them do. If they have a half-colored in shape, then that means they are carriers of that gene, or heterozygous for that. And so this is a pedigree here that shows albino people. Okay, and so these two parents both have normal color, but one of their children has albinism. So that means that each one of the parents, since albinism is recessive, okay, each one of these parents is heterozygous for the albino condition. Their other 3 offspring can either be homozygous dominant or heterozygous. This one you know is homozygous recessive, because that boy ended up being albino. So here we have the second generation, okay, that we have the daughter here and the son that has one of the parents that's albino, and they have one of their offspring, so you know that they both have to be heterozygous as well. This one would have to be heterozygous, okay, because the mother, since she's homozygous recessive, can only donate a lowercase, the recessive allele, whereas the father we don't know whether he is homozygous dominant or heterozygous, but he passed on obviously that dominant allele to his son. You can track families over many generations like this. You refer to individuals in each row. This is the generation 1, generation 2, generation 3, and so you can identify each individual by using the Roman numeral for the generation and the Arabic numeral for their person. So this person would be 1, 2, and this one would be 2, 4. This one would be 3, 2. Like that. Here's a more complicated one, same kind of thing. It's really handy to see this. I don't know what this one is about, but it makes it handy to see whether something is sex-linked or not, because if you see a lot more males than females showing up with the condition, then it very likely is going to be a sex-linked trait. And we'll do more things with pedigrees in class. This concludes the notes for genetics.